Hello, I'm Dr. Stephen Pipe from the University of Michigan, and I'm going to give an update on research that was presented at the American Society of Hematology meeting in December 2021. This was entitled Relationship Between Endogenous Transgene Factor VIII Expression and Bleeding Events Following Valoctocogene roxaparvovec Gene Transfer for Severe Hemophilia A. This was a post-hoc analysis of the GENEr8 phase III trial. So what we are presenting in this research is the relationship between endogenous factor VIII expression and bleeding events in participants with severe hemophilia A after gene transfer. Now, valoctocogene roxaparvovec is an AAV-based gene transfer that allows endogenous factor VIII production. And in the phase III GENEr8 trial, 134 adult men with severe hemophilia A received a single infusion of Valrox, and they experienced factor VIII production, resulting in reduced bleeding over 52 weeks. The relationship between factor VIII and bleeding rates is well established in epidemiological studies of hemophilia A, but it's unknown if a similar relationship exists following gene transfer. So we sought to answer this question using data from this trial. Briefly, eligible participants had previously been receiving standard of care factor VIII prophylaxis for over a year. And then they received a single infusion of gene therapy, and factor VIII prophylaxis was continued through 4 weeks post-infusion. We measured factor VIII activity with both chromogenic and one-stage assays, and bleeds were self-reported by the participants. And from the end of the prophylaxis up to the data cutoff date, 70 participants reported any bleed, and 30 participants reported having had a treated bleed. We then looked at the annualized bleeding rate for participants with low factor VIII activity at the end of year 1, as measured by both chromogenic and one-stage assay. As a group, the median ABR for these participants was in line with that reported from epidemiological studies of people with moderate hemophilia, so less than 5%. After gene transfer, the majority of bleeds were traumatic and located in joints. The analysis censored data for 1 month after discontinuing prophylaxis, and this was to allow for a transition period and to more accurately assess the relationship between the transgene-derived factor VIII and their bleeds. The median ABR for those with moderate hemophilia is similar to the median ABR that was observed for those who had low factor VIII activity at week 52. The relationship, interestingly, between the chromogenic factor VIII level and joint bleeds is similar to that reported for endogenous factor VIII level and joint bleeds in epidemiological studies of hemophilia A, suggesting that maybe this assay may be closer to being predictive of clinical breakthrough bleeding events. Of 134 participants, 5 of them had an ABR increase of at least 1 more bleed per year post-gene transfer compared to when they were on standard of care prophylaxis with factor VIII infusions. So we looked a little bit deeper into these 5 participants. The factor VIII by chromogenic assay was in the mild range for 3 of these, and most of their bleeds were due to trauma. Factor VIII was low throughout the study for the other 2. However, these participants did not choose to return to prophylaxis. In addition to these, there were 2 participants who did choose to return to prophylaxis. That was despite improvement in their ABR relative to their baseline values while on factor VIII prophylaxis. And after resuming prophylaxis, both of them had lower infusion rates and ABRs than they had prior to gene transfer, suggesting some impact from the endogenous transgene expression. So overall, we found a similar relationship between endogenous factor VIII and treated joint bleeding exists for participants in our trial, as in epidemiological studies of hemophilia A. So thus, this study gives additional confidence in the clinical correlates of factor VIII expression levels and expected bleed control.